Hi, this is our Osmosis Lab presentation with
Ariana Soar.
Ben Wilder.
and Felipe Cisneros. So, what is osmosis? Osmosis is the diffusion of water across a selectively permeable membrane. When a solution is isotonic, there is no osmotic flow across the membrane, as there are equal water molecules. When a solution is hypotonic, there is osmotic flow into the cell, as there are less water molecules in the cell than in the solution. When a solution is hypertonic, there is osmotic flow out of the cell as there are more water molecules in the cell than in the solution. One example of osmosis in our bodies is how it works in our kidneys. To balance out the salt and pH levels in the body, water moves in and out of the kidney. This is an important function of our body, and without osmosis, we wouldn't be alive.
One of the questions we had to answer for our osmosis experiment was, if an egg gains weight as it soaks, is the soaking solution hypertonic, hypotonic, or isotonic relative to the original solution? We believe the soaking solution is hypotonic because there is less solute in the egg than in the solution, so the egg will gain weight as it fills with the solution. Our next question was, what about when the egg loses weight? If the egg does lose weight, will the percent change in the egg mass be a positive or negative number? If the egg loses weight, it would be a negative number. Our hypothesis for the egg experiment was, if the egg is soaked in a solution with higher salt concentration, then the egg will lose weight. If the egg is soaked in a solution with a lower salt concentration, then the egg will gain weight. Here is some of the data of the sample recorded weights of the egg experiment. Here's the data of the egg's percent change in mass after being in the solution. We believe the solution with 0% is hypotonic, the solution with 7% is isotonic, and the solutions with 14% and 21% are hypertonic. For our gummy bear experiment, our hypothesis was, if you put a gummy bear in different solutions with different levels of solute, then you will be able to gather evidence and predict what solutions are hypertonic, hypotonic, or isotonic. Our method for the gummy bear experiment was to have 4 different gummy bears of the same color and put them into 4 different solutions. Each solution will have a different level of salt concentration. Then we will record the weights of each gummy bear before and after being soaked in the solution. Lastly, we will compare their weights to determine if the solution was isotonic, hypertonic, or hypotonic.
The materials needed for the experiment were 4 different gummy bears, same color from Black Forest Gummy Bears Company, 4 glass bowls filled with half a cup of water, salt measured in different amounts of tablespoons, a ruler to measure length and width, and a scale to measure the weight of gummy bears. The procedure and steps for this experiment were to fill 4 glass bowls with water, half a cup, measure weight and length and width of 4 orange gummy bears before putting them into the solution, measure different amounts of salt for each bowl, 1 bowl with no salt, 1 bowl with half a tablespoon, 1 bowl with 1 tablespoon, and 1 bowl with 2 tablespoons of salt. Then we would put 1 gummy bear in each bowl. After each hour, over a course of 3 hours, we would measure the gummy bears by measuring their weight, length, and their width. We would record the data each time we measured. After 12 hours, we would measure the gummy bears again by their weight, length, and width, and record the data. Here are some pictures of our experiment before the gummy bears were put into the solution. Here is our data collected on the weight of the gummy bears. As you can see, only 1 gummy bear gained and maintained more weight than the others. The other 3 gummy bears shrunk more or stayed relatively the same size. Here are more pictures of us measuring the gummy bears. Here are gummy bears' length, and this is how we recorded it during the experiment. Here are some pictures closer to the end, and it's interesting to see how discolored and dissolved some of the gummy bears got. Here is some more data on measuring the gummy bears and the width. And finally, these are some pictures of the gummy bears at the end of the experiment. So, discussing the experiment and our findings. It was interesting to see how the different solutions impacted the gummy bears. A lot of them changed colors, maybe looked bigger or smaller, and some fell apart closer to the end, kind of disintegrated. Only one gummy bear got larger and grew in size as the experiment continued. This gives supporting evidence that the first gummy bear was in a hypotonic solution. Gummy bears samples 3 and 4 had a very different appearance and size compared to gummy bear 1. This gives supporting evidence that the gummy bear samples 3 and 4 were more likely in a hypertonic solution. Only gummy bear sample 2 fell apart by the end of the experiment.
So, by having gummy bears placed into different solutions, we were able to gather more supporting evidence about the different types of solutions. If the solution had more solute than the gummy bear, then the gummy bear would gain weight. If the solution had less solute than the gummy bear, then the gummy bear would shrink, and that would give supporting evidence that it was in a hypertonic solution. If the solution had equal amounts of solute as the gummy bear, then the gummy bear would stay the same.
Thank you.